Hi, in this video I'm going to talk about cell cycle regulation. So we're going to start with G1. G1 is basically, it means gap 1. So this is when the cell had just got out from division. So basically the cell size is smaller than the normal size of the cell. So what it's actually trying to do, it will go for growth. So there will be growth factors in this phase, and there will be also any kind of detection for DNA damages or anything like that. So basically there will be a mitogen, which is a ligand that binds to a receptor, and this mitogen is basically a growth factor. So it's a growth factor. This is the signal where, okay, right now you need to grow more. So it goes and bind to the a receptor on, and then it will go inside of the cell until it reaches to so it will go activate RAS, and then RAS would activate MEK, and then this is a kinase, and then it will keep going until it reaches to the MYC gene, which is right here. So this is the MYC gene, which is this one right here. And this gene is specific for cellular growth. So once it's activated, it will actually keep going and say, okay, we're gonna grow right now. What's gonna happen next? It will activate cyclin D and cyclin E, which are these one. So this is E and this is D. And what those two are actually doing, they would be binding to CDKs, which are kinases. So E would bind to CDK 2, and it will activate it. And for D, it could actually bind to two CDKs, CDK 4 and CDK 6. Okay, so CDK 4 and CDK 6 is actually responsible for phosphorylating this part right here. So this part right here is the most important part in the G1. This is basically the restriction point, where if it's crossed, then it's also called commitment point. When when it's crossed, it could actually go to the S phase. So it's like a permission step. So this is BRB, which is retinoblastoma protein. And as you can see here, there is a blue part inside. This blue part is actually a co-transcriptional factor, which is called E2F. Okay, so what CDK4 and CDK6 do, they both go and phosphorylate this part right here to open up for the co-transcriptional factor, which is E2F, and go to bind on the regulatory sequence. Okay, where the cell would say, okay, we're gonna go to the next step, which is the S phase. So this is the permission for them to go to the S phase. After that, in the S phase, right now we crossed the S phase. We're in the S phase. Here, the DNA replication will begin. So before there was no replication, but right now there is a DNA replication. By the way, G1, in this step, the cell could actually go for days and being in this step, because this step actually has its commitment point, which is the BRB step right here. But for the rest, if we're gonna talk about S and G2, they are both they happen from hours to seconds. It doesn't last longer. Okay, so DNA replication has started, and in this step, it's regulated by the same CDK2 from from our first step, which is in G1. This one here, I colored it by the same color. So CDK2, right now it's not activated by cyclin E. Cyclin E is basically degraded, and also cyclin D and cyclin D is degraded also. So whenever they are done with their job, they get degraded. They are in continuous synthesis and degradation throughout the cell cycle whenever they are needed. So right now, this is a cyclin A, which bind to CDK2 and it activates it so that it goes to the next step, which is the G2 step. Now in G2 phase, basically chromatin is being regulated for mitosis. As you know, it's so right now in this phase, if chromatin is being regulated, that means it prefers the aochromatin where it's basically possible for transcription. So it will go through the process of acetylation of histone, where it opens up the parts where it's needed to be transcribed for the mitosis to occur after that. And also we have the cells builds up, and also we're having DNA replication still occurring. So and there is also a regulatory CDK in this process, which is CDK1, this one here in blue, CDK1, and it binds to cyclin B. Okay, and this CDK, this kinase, is regulated by, so if we're gonna say it's activated by CDC 25 and inhibited by WEE1. So in order for the cell to enter from the G2 phase to the M phase, which is right here, it has to go through this. CDK1 has to be activated in order to enter the M phase. And the reason why it has to be there, for that CDK1 has to be there, is because CDK1 is basically, it could code for P34, which is responsible for phosphorylating of lamina. So lamina is it's an exclusive intermediate in the nucleus. So when you phosphorylate lamina, if there are a lot of phosphate group in lamina, it will just be it will disappear. So when we're doing the division, we don't want any kind of cell adhesions or cytoskeletons, so that it doesn't get into the separation process. So in order to degrade these cytoskeletons, it's phosphorylated by this particular gene, which is CDK1, and also it's called also CDC2. Both of them, those are genes. So the code for P34, which phosphorylates lamina. So right now the cell could freely separate the chromosomes from each other. So once we're reaching here, also here there is a regulatory step in metaphase. So in metaphase, this is when the chromosomes are aligned together. So right now we want to make sure that every single thing is okay, and this is done by anaphase promoting complex. So it's APC, anaphase promoting complex. So what it does, it actually makes sure that every single thing is okay, and also it degrades all all of the other cyclins, which in this case we're having here cyclin B, it's gonna be degraded from here. So cyclin B will be degraded and the separation process will be initiated in the anaphase. And both of them are gonna be separated from each other and it will occur correctly. So there are general inhibitors in here. So these inhibitors are basically, they are stimulated whenever there is a stress on the DNA or there is stress going in the cell or there is a damage, a particular damage or anything like that. So these cells are basically, or these kind of genes are activated to make the cell go through apoptosis, which is the cell programmed death. So these inhibitors, we're having CIP, KIP family. These one here are P21, P27, and P57. And basically what they do, they inhibit CDK2. Okay, remember CDK2 was important in G1 and also it's important in the S phase. It's important in both of them. So they inhibited, and they just, whenever the cell stays too long in the S phase, it will just be there will be determination that it will go through apoptosis or cell death. It's not allowed for the cell to stay too long in the S phase or the G2 phase, but it's okay to stay long in the G1 because this phase here got the commitment point. Also, we're having the ENC4 family. This family is P14, P16, P18, and P19. Specifically for P16, it inhibits CDK4 and CDK6. Okay, CDK4 and CDK6, they are so essential for G1. And also, they are so important for phosphorylating the retinoblastoma protein, which is the restriction point that lets this is what gives the permission to go to the S phase. So without this CDK4, CDK6, you will never be able to get to the S phase, and eventually that will suppress the cell growth. And also, one of the most important tumor suppressors or inhibitors of the cell cycle is P53. So it's actually regulated by the ATM ART gene. These basically code for ATM ART kinase, and what this does, it could actually activate the P53 via phosphorylating. So the P53 and the cell exists like so. So this is the P53. Attach it to it MDM2. So whenever the ATM ART kinase is active, then we'll go there and bind and phosphorylate it. So it's phosphorylated right now. This part right here will be dissociated, so it will P53 will just be free. So it will go and activate the gene responsible for P21. And as you know, as I just mentioned, P21 is one of the CIP, KIP family. It inhibits CDK2. Okay, and this is how it regulates the cell cycle. It's a very common tumor suppressor in cancer.